Welcome back to the deep dive. Today we are really going deep. We're tackling one of the most fundamental systems in the body.
Absolutely. We're moving beyond just basic biology here.
We're talking about the endocrine system, those chemical messengers, the hormones that control, well, practically everything: growth, stress, metabolism, our existence, really.
And in this deep dive, it's really geared towards achieving, let's say, professional mastery. Our goal here is synthesis.
Synthesis, how so?
Well, we're taking all this complex information, the physiology, the pharmacology, and weaving it together from the source gland right down to how each hormone actually works at the cellular level.
So not just what they are, but precisely how they run the show.
Exactly. Understanding the mechanism is key to that mastery level.
Okay, and that starts with the name itself, doesn't it? Hormone. It comes from the Greek hormone.
That's right, to stir up, which is, I think, a perfect description. These aren't gentle suggestions. They are powerful signals.
Powerful signals produced by specialized cells traveling through the bloodstream.
And hitting very specific targets far away. That's the essence of endocrine action.
And you mentioned synthesis. What are the core functions we need to synthesize here? What do hormones fundamentally do?
Broadly speaking, two critical things. First, they orchestrate the big, programmed life events. Think development, puberty, reproduction, metabolism over years, even aging. It's the long game.
The body's long-term plan, laid out by hormones. Okay, what's the second function?
The second is maybe more immediate, more constant. It's maintaining homeostasis, keeping the internal environment stable.
Right, that steady state, regardless of what's happening outside or even inside, like after a meal.
Exactly. Temperature, blood sugar, fluids.
Yeah.
All of it needs to stay within tight limits. Hormones are the key adjusters, providing that stability, sometimes rapidly, sometimes over the long haul.
Okay, let's unpack this. Let's dive into the specific jobs. You said there are 7 major areas hormones control. If we get these, we get the whole picture.
That's the framework. Master these 7, and you've got a solid handle on endocrinology. So first up, maybe the most obvious one, fuel availability, managing energy, glucose, fats, deciding what to use, what to store.
Ah, okay. And that immediately brings insulin and glucagon to mind, right? The classic pair.
The classic antagonists, yeah. Insulin pushes glucose into cells for storage or use, lowering blood sugar. Glucagon does the opposite, pulls glucose out of storage, raising blood sugar. It's that minute-by-minute balancing act.
But it's not just them, is it? You mentioned growth hormone earlier in a different context.
Good point. Growth hormone is also involved here. We think of it for growth, obviously, but it's a major player in metabolism, too. It affects how we handle glucose and lipids over the longer term. So it's part of energy planning, not just the immediate response.
Okay, so insulin, glucagon for the short term, growth hormone for the longer view on fuel. Got it. What's next?
Number 2, metabolic rate. This is the overall speed of the body's engine. How fast and efficiently are we burning that fuel?
And this is squarely in the territory of the thyroid hormones.
Exactly. Triiodothyronine, which is T3, and thyroxine, T4, they set the pace for almost every cell.
And there's a key difference between T3 and T4, isn't there?
T4 is produced in larger amounts, but it's more of a storage form, a pro-hormone, really. T3 is the one that's much more biologically active. The level of T3 dictates your basal metabolic rate, how much oxygen you consume, even how much heat you produce.
Like the body's thermostat and accelerator pedal combined?
Yeah.
Okay. Third function.
Somatic growth, building the physical body.
Growth hormone's main job, right?
Well, yes and no. GH kicks things off, but it works indirectly. It mainly stimulates the liver.
To produce what?
To produce insulin-like growth factors, or IGFs. It's mostly the IGFs that actually drive the cell division and differentiation needed for bones, muscles, for actual physical growth.
Ah, so GH is the command, but IGFs are the workers on the ground. It's a partnership.
It's a crucial axis, yes. Highly coordinated.
Okay, number 4. This one covers a huge part of life. Sex and reproduction.
Absolutely. Everything from the changes at puberty to fertility cycles in adults, maintaining male and female characteristics, it's all hormone-driven.
And the key players here are the gonadotropins.
Right. FSH, follicle-stimulating hormone, and LH, luteinizing hormone. They come from the pituitary and act on the gonads, the testes, and ovaries.
Prompting them to produce the sex steroids.
Exactly. The androgens, like testosterone, the estrogens, like estradiol, and the progestins, like progesterone. And the way FSH and LH are released, often in pulses, is also critical, especially for fertility. It's complex timing.
Okay, 5th function. This sounds more like plumbing. Circulating volume, fluid balance, electrolytes, blood pressure.
Directly linked to blood pressure, yes. And it's tightly connected to kidney function. two main hormones here.
Which are?
Aldosterone's mineralocorticoid makes the kidneys hold on to sodium and water and get rid of potassium.
Okay, so aldosterone manages salt and water together. What's the other one?
Antidiuretic hormone, ADH, also known as vasopressin. ADH is more about pure water balance. It tells the kidneys to reabsorb water, making urine more concentrated. Helps keep you hydrated without necessarily changing sodium levels as much as aldosterone does.
Got it. Aldosterone for salt, ADH for water. Okay, number 6. Adaptation to stress. How the body reacts when under pressure.
And there are two phases to this, really. The immediate O response and the longer-term coping mechanism.
Right, the immediate jolt comes from.
Adrenaline from the adrenal medulla. That's your fight or flight. Heart pounds, blood pressure spikes, fuel mobilizes instantly.
Okay, that's the sprint. What about the marathon of stress?
That's where the glucocorticoids come in. Hormones like hydrocortisone or cortisol from the adrenal cortex.
And they do what during stress?
They manage things longer term. They adjust inflammation, modulate the immune system. You don't want it overreacting during a crisis. And crucially, they make sure the brain and muscles have a steady glucose supply. They help the body endure prolonged stress.
2 distinct stress axes. Fast adrenaline, sustained cortisol,
Okay.
and the final function, number 7.
Calcium balance. Keeping the levels of this crucial mineral incredibly stable.
Why is calcium so important?
Oh, it's vital for nerve function, muscle contraction, blood clotting, bone health, lots of things. The body keeps it in a very narrow range.
And how does it do that? 3 main players you mentioned.
Yep. The main one raising calcium levels is parathormone, or PTH, from the parathyroid glands. It acts on bone, kidneys, and gut to boost calcium in the blood.
Okay, PTH raises calcium. Who lowers it?
That's calcitonin, which actually comes from the thyroid gland. It generally opposes PTH, helping to lower blood calcium.
And the third player?
Vitamin D. Specifically, it's active form calcitriol. You need a vitamin D to effectively absorb calcium from your food. So PTH, calcitonin, and active vitamin D work together in this constant feedback loop.
Wow, 7 huge jobs, all managed by these chemical signals. The complexity is mind-boggling.
It really is. Which makes understanding the sources even more important.
Okay, so let's map it out. Where do these messengers actually come from? This is where it gets really interesting. Locating the command centers.
Right. We need that anatomical grounding.
Yeah.
Start at the top. The control tower. The pituitary gland. Sits right under the brain.
And it's not just one thing, is it? It's divided.
Correct. Anterior and posterior pituitary. They look close, but they have totally different origins and functions.
Okay, let's tackle the anterior pituitary first. That's the one making a lot of hormones that control other glands, right? The trophic hormones.
Exactly. 6 major ones we need to know.
Lay them out for us.
Okay. First, growth hormone, GH. We've
Yeah.
talked about its role in growth and metabolism. Then prolactin, PRL. Main job is stimulating milk production.
Got it. GH. Prolactin. What else?
Adrenocorticotropic hormone, AZTH. The name tells you. It targets the adrenal cortex, also called corticotropin.
Makes sense. ACTH controls cortisol release.
Precisely. Then thyroid-stimulating hormone, TSH, or thyrotropin. Again, the name gives it away, controls the thyroid gland.
Okay, that's 4. What are the last 2?
The gonadotropins. They act in the gonads. There's follicle-stimulating hormone, FSH, and luteinizing hormone, LH. Both are key for reproduction.
6 hormones, mostly commanding other glands. That's the anterior pituitary. What about the posterior pituitary?
It's different. It doesn't actually make hormones. It stores and releases 2 hormones that are made up in the hypothalamus in the brain.
Ah, okay. So it's more like a storage depot. Which hormones?
Oxytocin, famous for roles in childbirth, lactation, social bonding. And the other one we already mentioned, antidiuretic hormone, ADH, or vasopressin, the water regulator.
So anterior makes and releases. Posterior just stores and releases hypothalamic hormones. Clear distinction. Okay, moving down from the pituitary.
Down the neck, we find the thyroid gland. We know it makes the metabolic regulators T4, thyroxine, and T3, triiodothyronine.
And didn't you say it makes calcitonin too?
Yes, the calcium-lowering hormone. So T3, T4, and calcitonin from the thyroid. And tucked right behind or within the thyroid are the tiny parathyroid glands.
And they make...
Parathyroid hormone, PTH, or parathormone, the main calcium-raising hormone.
Okay. Thyroid and parathyroid covered. What's next on our tour?
Let's go to the pancreas. Not the whole pancreas, but specific clusters of cells called the islets of Langerhans.
Right, the endocrine part of the pancreas. And they make...
The big 2 for fuel management. Insulin from beta cells and glucagon from alpha cells. Released right into the blood to control glucose.
Perfect. Now, the adrenals, sitting on top of the kidneys, right?
Right.
And they're also split.
Yes, a crucial split. Outer layer is the adrenal cortex. Inner part is the adrenal medulla. Completely different functions.
Let's do the adrenal cortex first. That's the steroid factory.
Exactly. It makes hormones derived from cholesterol.
Mm-hmm.
And it has zones, each making different types. You've got glucocorticoids. Cortisol hydrocortisone is the main one for stress and metabolism.
Okay, glucocorticoids. What else?
Mineralocorticoids, primarily aldosterone for salt and water balance.
Right, we covered aldosterone. So glucocorticoids, mineralocorticoids, anything else?
And some sex steroids. Things like DHEA, dehydroepiandrosterone, which are precursors that can be converted to androgens or estrogens elsewhere.
So the cortex makes 3 classes. Sugar, gluco, salt, mineralo, and sex steroids. Easy way to remember it.
That's the classic mnemonic, yeah.
Mm-hmm.
Now, the adrenal medulla, the inner part.
That's the fast-acting one.
Yes, it's really part of the nervous system, functionally. It pumps out adrenaline and noradrenaline for that immediate stress response.
Got it. Cortex for slow steroids, medulla for fast adrenaline. Okay, where next?
The gonads. Tests in males, ovaries in females.
And they produce the primary sex steroids.
Correct. Testes produce androgens, mainly testosterone. Ovaries produce estrogens like estradiol, and also progestins, primarily progesterone. All essential for reproduction and secondary sex characteristics.
Okay, pituitary, thyroid, parathyroids, pancreas, adrenals, gonads. Are those all the major glands?
Those are the main dedicated endocrine glands, but we also need to mention non-glandular or temporary sources. The prime example is the placenta during pregnancy. It's a temporary organ, but it's an absolute hormone factory.
Really? What does it make?
A whole suite of things needed to maintain the pregnancy and support the fetus.
Mm-hmm.
Chorionic gonadotropin, HCG, that's what pregnancy tests detect. Also prolactin, lots of estrogens and progesterone, something called placental lactogen, even chorionic thyrotropin.
Wow. So the placenta temporarily takes over or adds to many endocrine functions.
It has to. It's coordinating the mother's physiology to support a whole other organism. It's a fascinating temporary endocrine powerhouse. Okay, so we've mapped the glands. But if we zoom out, remember the pituitary is often called the master gland.
But it takes orders from higher up, right?
Exactly. The hypothalamus, which is part of the brain, is the real supervisor, especially for the anterior pituitary. It connects the nervous system, thoughts, feelings, environmental cues to the endocrine system.
And it does this using its own set of hormones, the releasing and inhibiting factors.
Precisely. These are crucial. They travel just a tiny distance through a special portal blood system directly to the anterior pituitary to tell it what to do.
This hypothalamic pituitary axis, understanding these specific signals feels like the next level of mastery. You mentioned their chemical nature is important.
Very important. It affects how they work and how we can potentially use them or block them therapeutically. Let's break them down. First, the releasing hormones, the ones that stimulate the pituitary.
Okay, who's first?
Thyrotropin-releasing hormone, TRH. It tells the pituitary to release TSH. Chemically, it's tiny, just a tripeptide, 3 amino acids.
TRH, a tripeptide, stimulates TSH. Got it.
Next, corticotropin-releasing hormone, CRH, tells the pituitary to release ACTH. This one's quite a bit bigger, a peptide of 41 amino acids.
TRH, 41 amino acids, stimulates ACTH.
Okay. Then for reproduction, gonadotropin-releasing hormone, GnRH, also known as LH, RH, FSH, HRH, or gonadorlin. It stimulates the release of both FSH and LH. It's a decay peptide, 10 amino acids.
GnRH, decay peptide, stimulates FSH and LH.
Right.
And the last releasing hormone.
Growth hormone-releasing hormone, GHRH, stimulates GH release. It's another peptide found in forms with 40 or 44 amino acids.
GHRH, 40 or 44 amino acids, stimulates GH. Okay, those are the 4 main stimulators. Now, what about the brakes? The inhibitory hormones.
2 key ones here. First, the one that inhibits prolactin release. Prolactin release inhibitory hormone, PRIH.
Okay, and what's special about PRIH?
Chemically, it's unique on this list. PRIH is actually dopamine. It's not a peptide, it's a monoamine, a much simpler molecule.
Wow, okay, so TRH, CRH, GnRH, GHRH are all peptides of varying sizes, but PRIH is dopamine. That seems significant.
It's hugely significant. Think about drug development. It's much easier to make an orally active drug that mimics or blocks a small molecule like dopamine than it is for a large, fragile peptide hormone that would get digested.
Ah, that makes sense. That's why dopamine agonists are common treatments for high prolactin levels. They can be taken as pills. You couldn't easily do that with a peptide like GHRH.
Exactly. That chemical difference has major practical implications. Okay, the second inhibitory hormone.
What is it?
It's somatostatin, also known as growth hormone release inhibitory hormone. So it damps down GH secretion. It's a peptide, 14 amino acids long.
Interesting. So growth hormone is unusual in having both a dedicated releasing hormone, GHRH, and a dedicated inhibiting hormone, somatostatin.
Yes, it points to the need for really fine-tuned control over growth and metabolism. It's not just on or off. There's a constant balance between stimulation and inhibition.
Okay, so we have the hormones, their sources, their controllers. Now the million-dollar question: what actually happens when a hormone reaches its target cell? This is where the pharmacology really kicks in, right? The signaling language.
Precisely. And the good news is, despite the huge number of hormones, there are really only three main ways they deliver their message, three dialects, if you will. And it all depends on where the hormone's receptor is located.
Location, location, location. Okay, what's the first location?
The cell membrane. This is typically for hormones that can't easily pass through the fatty cell membrane, mainly the protein and peptide hormones, and also adrenaline.
And because the receptor's on the outside, the signal needs to be relayed inside.
Exactly. They use intracellular second messengers. This usually leads to rapid effects, often easily reversible.
Okay, what's the first major pathway involving these membrane receptors?
The most common one is via alteration of intracellular CMP concentration, cyclic AMP.
How does that work? Walk us through it.
Okay. Hormone binds to the receptor outside. This activates a G protein linked to the receptor inside. The G protein then turns on or off an enzyme called adenyl cyclase.
An adenyl cyclase makes a CAMP.
Right, or stops making it. So the level of KMP inside the cell changes dramatically. CMP then acts as the second messenger.
What does CAMP do?
It usually activates another enzyme called protein kinase A, pKa. pKa then goes around adding phosphate groups to other proteins, changing their activity. This cascade amplifies the original signal enormously. One hormone molecule outside can change thousands of molecules inside.
Amplification. Got it. Which hormones use this CAMP pathway? Seems common.
Very common. Think adrenaline, glucagon, TSH, FSH, LH, ACTH, many of the pituitary hormones, PTH, calcitonin. Even some of those hypothalamic releasing hormones use it. It's a workhorse pathway for fast responses.
Okay, CMP is one major second messenger system. What's another way membrane receptors can signal?
The second key pathway is via IP3D gene generation. This one involves calcium ions.
IP3 and DDG, what are they?
When the hormone binds, a different G protein activates an enzyme called phospholipase C. This enzyme cleaves a membrane lipid into two second messengers, inosityltrisphosphate, IP3, and diacylglycerol, DEG.
Two messengers from one reaction. What do they do?
IP3 floats over to the endoplasmic reticulum, the cell's internal calcium store, and opens channels, releasing calcium, Ca2+, into the cytoplasm. DEG stays in the membrane and activates another enzyme, protein kinase C.
PKC.
So this pathway boosts intracellular calcium and activates PKC. Which hormones use this? Good examples are vasopressin, ADH, acting on its V1 receptor, the one that causes blood vessel constriction and oxytocin.
Wait, you specified the V1 receptor for vasopressin. Does it use a different pathway for its water-retaining effect in the kidney?
Excellent point, yes. The V2 receptor for vasopressin, found in the kidney tubules, typically uses the CAMP pathway we just discussed. That pathway leads to the insertion of water channels, causing water reabsorption.
Ah, so the same hormone can use different signaling pathways in different tissues via different receptor subtypes.
Exactly. V1 uses IP3DAG for rapid vasoconstriction. V2 uses CAMPT for the slower, more sustained effect on water balance. That's a critical level of detail for mastery. Sometimes in the V2 pathway, the calcium release later acts almost like a third messenger.
Fascinating. Okay, CMP and IP3DAG.
Yes.
Some receptors have their own built-in enzyme activity. This is direct transmembrane activation of tyrosine protein kinase.
So the receptor itself is an enzyme.
Pretty much. The part inside the cell is a tyrosine kinase. When the hormone binds to the outside part, the inside part gets activated and starts phosphorylating tyrosine amino acids on itself and other target proteins. Phosphorylation again, but directly by the receptor. No G protein middleman needed here.
Right. It triggers a phosphorylation cascade, activating downstream signaling pathways and enzymes directly. It's a very powerful signal.
Which hormones use this direct kinase activation?
This is characteristic of hormones involved in growth and metabolism. Think insulin, growth hormone, and prolactin. Their receptors work this way. It makes sense for hormones that need to orchestrate complex long-term cellular changes.
Okay, three membrane mechanisms, CMP, IP3DAG, and direct tyrosine kinase, all relatively fast-acting via second messengers or direct enzyme action. What's the second major location for receptors?
Inside the cell in the cytoplasm. This is where the lipid-soluble steroid hormones at.
Steroids, like glucocorticoids, mineralocorticoids, sex hormones.
Exactly. And because they're fatty, they can slip right through the cell membrane easily. No need for a surface receptor.
So what happens once they're inside?
They find and bind to their specific receptor protein, which is just waiting in the cytoplasm. This binding causes a change in the receptor shape.
What does that shape change do?
It unmasks a part of the receptor that can bind to DNA. The whole hormone receptor complex then travels into the nucleus.
Into the command center. And then?
It binds to specific sequences on the DNA, called hormone response elements. This binding directly influences gene transcription, the process of making messenger RNA, mRNA, from a gene.
So it's turning genes on or off.
Essentially, yes. It leads to increased or decreased synthesis of specific mRNA molecules, which then get translated into new functional proteins. These new proteins are what actually change the cell's behavior.
So the effect isn't immediate. You have to wait for the genes to be transcribed and new proteins to be made.
Precisely. That's why steroid hormone effects like muscle growth from testosterone or the anti-inflammatory effects of cortisol take hours or even days to fully develop. It's a slower but more profound and lasting change based on altering protein synthesis.
Makes sense. So the steroids acting via cytoplasmic receptors are glucocorticoids, mineralocorticoids, androgens, estrogens, progestins, any others?
Yes, one more key player acts this way: calcitriol, the active form of vitamin D. Even though it's not strictly a steroid, it's lipid-soluble and works through a similar cytoplasmic receptor mechanism to control genes involved in calcium transport.
Good to know. Okay, that's membrane receptors and cytoplasmic receptors. What's the third and final location?
Also inside the cell, but specifically nuclear receptors. This mechanism is unique to the thyroid hormones.
T3 and T4, how is this different from the steroids? They're also fairly small and lipid-soluble, aren't they?
They are, and they also get inside the cell easily. But their receptors aren't waiting in the cytoplasm. The thyroid hormone receptors are typically located already inside the nucleus, often bound directly to the DNA even before the hormone arrives.
So the hormone goes straight into the nucleus?
Yes, it enters the nucleus, binds to its receptor there, and this binding then alters gene transcription, similar to steroids, leading to changes in protein synthesis.
So both steroids and thyroid hormones work by changing gene expression, leading to slow genomic effects. But the key difference is where they first meet their receptor. Cytoplasm for steroids, nucleus for thyroid hormones.
That's the core distinction. Both result in altered protein synthesis, explaining their profound, long-lasting effects on things like metabolic rate, thyroid, or inflammation steroids. But the initial binding site differs. So, having understood all this, the sources, the mechanisms, the different receptor types, how do we actually use this knowledge in medicine? Endocrinology provides a huge number of therapies.
Right, it's not just academic. How is this applied? What's the most basic application?
The most straightforward is substitution therapy. If the body isn't making enough of a hormone, we simply replace it.
Like giving insulin to someone with type 1 diabetes or thyroid hormone for hypothyroidism.
Exactly. We use either the natural hormone or quite often a synthetic analog, a slightly modified version to restore normal levels.
Why use synthetic analogues instead of just the natural hormone?
Sometimes they work better therapeutically. Maybe they last longer in the body, so you need fewer doses. Or perhaps they have better absorption if taken orally, though that's tricky for peptides, as we discussed.
Yeah.
Or maybe they're more stable. For instance, we usually give synthetic T4-L-thyroxine for hypothyroidism because it has a long, stable half-life, providing steady levels, rather than the more potent but shorter-acting T3.
Okay, so substitution is replacing what's missing. What's another therapeutic approach?
Pharmacotherapy. This is where we use a hormone, or often a synthetic analog, not just to replace it, but to achieve a specific drug effect, often at doses much higher than the body would ever produce naturally.
Like using steroids not for adrenal failure, but for their other effects.
Exactly. The classic example is using high-dose glucocorticoids like prednisone or dexamethasone. We're not treating a cortisol deficiency. We're harnessing their powerful anti-inflammatory and immunosuppressive actions.
To treat things like severe asthma, rheumatoid arthritis, or prevent organ transplant rejection.
Precisely. We're exploiting that slow genomic action on protein synthesis to shut down inflammation pathways throughout the body. It's using the hormone as a drug for a purpose beyond simple replacement.
Makes sense. Substitution and pharmacotherapy. What's the third angle? Sometimes we want to stop hormonal action, right?
Absolutely. That involves using antagonists and inhibitors. This is critical when a gland is overactive. or when we need to block the effects of excess hormone.
How do antagonists work?
Hormone antagonists are drugs that bind to the hormone's receptor but don't activate it. They just sit there, blocking the natural hormone from binding and doing its job, like putting the wrong key in a lock.
Can you give an example?
Sure. In some types of prostate cancer that are driven by testosterone, we use anti-androgen drugs that block the testosterone receptor.
Okay, so antagonists block the receptor. What about inhibitors?
Synthesis or release inhibitors work earlier in the process. They actually stop the body from making the hormone in the first place or stop the gland from releasing it.
Like drugs that block cortisol production in Cushing's syndrome. Or those dopamine agonists we talked about that inhibit prolactin release.
Exactly. So you can block the receptor with an antagonist, or you can stop the hormone being made or released with an inhibitor. Both are crucial strategies when dealing with hormone excess.
So we can amplify effects with high doses, or we can block or inhibit. That covers a lot of therapeutic ground. #tag outro.
So looking back, we've really covered the endocrine system from top to bottom for the meaning of hormone itself,
All the way through the glands, the hypothalamic control.
mapping out the specific hormones like the 6 from the anterior pituitary, the adrenal steroids, sugar, salt, sex.
And crucially, down to the 3 fundamental ways hormones act at the cellular level based on receptor location.
And that location piece is really the key takeaway for mastery, isn't it? If you know the receptors on the cell membrane.
You know the action is fast, seconds to minutes, using second messengers like CAN-AMP or IP3-DIG for amplification. Think adrenaline, glucagon, most pituitary hormones.
Right. Quick adjustments. But if the receptor is inside the cell, either cytoplasmic-like for steroids or nuclear-like for thyroid hormones,
Then you know the effect is slow hours to days because it involves changing gene expression, making new proteins. It's about fundamental, lasting changes.
that link between receptor location and the speed and nature of the response, rapid signaling versus slow genomic action, that's the core principle. It explains why some hormones fine-tune homeostasis moment to moment, while others orchestrate long-term development or adaptation.
Absolutely. Understanding that cellular geography is key to predicting and manipulating hormone effects therapeutically. Okay, so to wrap up, let's leave our listeners with something to think about, building on what we discussed.
Sounds good.
We highlighted the chemical difference between most hypothalamic releasing inhibiting hormones, which are peptides, and PRIH, which is dopamine. We noted GHRH is a fairly large peptide, 44 amino acids. So here's the thought. Given that difference, large peptide versus small monoamine, what specific practical challenges would researchers face in trying to develop an orally active drug that mimics GHRH compared to the relative ease of developing oral dopamine agonists or antagonists? Think about what happens to peptides in the digestive system versus small molecules.
That's a great question. It forces you to connect the chemistry we discussed directly to the real-world problem of drug delivery, something definitely worth mulling over.
Consider that challenge as you continue your own deep dive. Thanks for joining us today as we work towards mastering the endocrine system.
It was a pleasure. Until next time.
We'll catch you next time on the deep dive.